All right, our third lesson in the biochemistry unit on biomolecules is starting now. So we've talked about molecules and compounds, but there's a special word that certain molecules get. They're called macromolecules, which just means large molecules. These are considered to be chemicals called polymers. The word poly means many, and mer means unit. So this means many units. So essentially what polymers are molecules with a repeated basic structure. So they have a subunit or a basic look to their molecule, and then they just add that same look over and over and over and connect it to each other to make like a long chain. So the repeated basic structure is called a monomer. Mono meaning one. So the way I like to think about this is Legos. A monomer would be one single Lego. This Lego, though, can connect to another Lego, which can connect to another Lego and another Lego. Connecting them all together to build a bigger structure is what we'd be doing to make the polymer, to make our macromolecule. But all of the monomers are the same. They're all the same little piece, and we just add them together to make a bigger structure. Now, there's 4 macromolecules that we've termed biomolecules because they're essential to life. We find them in all living things. These are carbohydrates, lipids, proteins, and nucleic acids. So what are carbohydrates? You've probably heard this term before, mostly surrounding our food. All these will kind of sound familiar when we put them in terms of food. So carbohydrates, their basic definition is that they're a sugar or a quick energy source. They're made of molecules that contain the elements carbon, hydrogen, and oxygen. Now, their monomer, their basic unit or subunit, is called a monosaccharide, meaning one sugar. And it's going to look like this picture over here. The basic monomer actually has a name. We call it glucose. It's one of the most simple sugars out there, and it's usually the thing that ends up building on others to make bigger structures. Now, sometimes we connect 2 of these sugars together, 2 of these monosaccharides, to create a disaccharide. So it would just look like 2 of these little sugars connected to each other. And then when many of them are connected together, we call it a polysaccharide, many sugars. So carbohydrates are used in the body as a quick energy source. That's why if you've ever played sports, your coach may say you have to carb load the night before. So eat a big pasta meal or eat bread the night before your big game or race, because all of those carbs will give you a quick energy source to tap into when you're doing your competition. So examples of these are things like glucose, sucrose, fructose. You've probably heard a lot of these before. Notice that they all tend to end in OSE. That's just one of the things that scientists do to help us clue into, oh, this is going to be a carbohydrate. So on the test, this picture to the right, this is all I really want you to know. That it's made of carbon, hydrogen, and oxygen. Its monomer is glucose, or the monosaccharide. I would like you to know that term at least. It's used for quick energy, and it's found in things like breads and pasta, the foods that we would call carbohydrates. Now, lipids are kind of like carbohydrates' long-lost sibling. They're both made of mostly carbon and hydrogen with a little bit of oxygen. But these ones, instead of supplying you with quick energy, these supply you with long-term energy. We store energy in fats. And we also use these lipids to create special structures like cell membranes, the part of the cell that's going to surround all of our internal little organelles. So basically, these are fats, oils, and waxes, and they give us a long-term energy storage source. Their monomer, their subunit or their basic unit, is called a fatty acid. Now, there's 2 or 3 really different types of fatty acids that we're going to talk about here. Saturated fats, unsaturated fats, and trans fats. Now, the 2 natural ones are going to be saturated and unsaturated fats. So let's talk about those first. In a saturated fat, which is the top line here on our monomer, on our fatty acid, it has all single bonds connecting these carbons. And so because of that, it's going to look like a straight chain. So these are going to be solid oil, or they're going to be solid lipids at room temperature. So they're not oils, they're like fats or waxes. Things like animal fat or butter or ghee or coconut oil. Unsaturated fats have mostly single bonds, but every once in a while you'll find that they have a double bond. And this double bond creates a different shape than our saturated fats. So it's going to have like a little bend or a kink in it. And these are going to be liquid at room temperature. Things like olive oil or vegetable oil are your unsaturated fats. Now, you may have heard the term trans fat before. These are human-made fats. We take the normal fats and we modify them, and they're really bad for your health because they tend to stack up on top of each other and they can clog your arteries and your blood vessels. The last thing I wanted to mention with lipids is that they're also an example is a wax. So a wax is just a very special lipid that plants tend to make. And if you've ever gone and touched a leaf, you might feel that wax on it. Plants will make this to cover up the leaf so that they don't lose water. It's kind of like a waterproofing mechanism. So that way the plants don't have to work so hard to keep bringing water up through their roots and they don't lose precious water. So once again, make sure you know this for the test, this picture, that it's mostly carbon and hydrogen, but that it does have some oxygen, excuse me. These are fats, another term for them. They're made of glycerol and fatty acid tails. So these are the fatty acid tails, and this is the glycerol part. So it's just part of the monomer, which is a fatty acid. And it's used in energy storage and building things like cell membranes. So examples are butter and oil. Our next biomolecule is the protein. You've probably heard of it before when you talk about food. So the proteins that you eat are things like muscles from other animals. The basic definition for you guys is going to be that they're structural molecules that build up our bodies, excuse me. And there's a certain example that we're going to talk about, a certain type called an enzyme that's going to be used to speed up chemical reactions. They are also made of carbon, hydrogen, and oxygen, but also nitrogen. We add that into our mix of elements as well this time. So you can see our little nitrogen right there. Now, their monomer is going to be called an amino acid. And there's 20 different versions of these amino acids. This is the basic structure right here. But now this little R stands for a variable group. There is really no element called R. So what's going to go there is any one of these 20 different types, kind of like our alphabet. We have 26 different letters, and we can mix those letters in many different ways to form different words and sentences and paragraphs and have conversations. These proteins can be many different sizes and shapes and structures because these 20 different types can all bond with each other in different ways and different orders. Now, these amino acids are going to connect to each other through a special bond called a peptide bond. The way I like to think of it as, think of P for protein. It's going to be a protein bond. So these peptide bonds are going to link an amino acid to an amino acid. And as we do that, we get what seems to be this like big long chain of amino acids strung together, just like that. So we would call that a polypeptide, or many peptides. There's many of these peptide bonds holding them together. And eventually what's going to happen is this polypeptide is going to start to bend and twist and form this really complex shape that we know of as our protein. And the shape of the protein is so important because it determines how the protein works. There's something called denaturing. A protein can denature, and what this means is it will lose its shape if it's put in very harsh conditions. Proteins can only really work and stay the same shape at very specific pHs, at very specific temperatures. And if you make things too extreme, then they'll fall apart and lose their entire shape. So once a protein denatures, it completely stops working, and most of the time it will never work again. Most of the time this denaturing is permanent. And you've actually seen this happen before. If you've ever cooked eggs, that the egg itself goes from like clear to white. And what's happening there is the proteins in that egg are being denatured because of the high heat, and they're changing into a form that can never be put back the way it was. You can never unscramble that egg. So where you can find these are things like chicken, fish, eggs, all of our meat products essentially. So remember, know this part for the test. Know all of this, that it's made of carbon, hydrogen, and oxygen. Its monomer is an amino acid, and it has this R group here that determines which one of the 20 amino acids it's going to use, and that it's used up to build our structure of our bodies like muscle and bone, and then we have special enzymes that control our reaction rates. Our last biomolecule, woohoo! These are nucleic acids. You probably haven't heard them called this before, but you've probably heard of DNA. And DNA stands for deoxyribonucleic acid. So it's just one of our types of nucleic acids. Another one that you may have heard before is RNA. And that's something that we are going to talk about later in the year. We have DNA and RNA as our two main examples of a nucleic acid. So what are these? These are molecules that contain the code of life. And they contain elements carbon, hydrogen, oxygen, and nitrogen, just like everything before. We're building, but this time we're going to add one more, and that's phosphorus. You can see our little phosphorus right here. So the monomer for a nucleic acid is called a nucleotide. These nucleotides are made of a few main parts. So we'll have a phosphate group. We'll have a sugar here. You can kind of see that this looks like that overall structure of our carbohydrate. And then this is what we call a nitrogen base. It's got a lot of nitrogen in it. And you've probably heard of these nitrogen bases before, but not called such. You may have heard them called A, T, C, and G, the code for our DNA. So the sugar and the phosphate, these two parts here, make up the backbone of the DNA, this dark part going and twisting around on our DNA here. And the nitrogen bases make up these colorful rungs of the ladder, the connections between. And those bases contain the code of DNA. So remember to learn this part for the test. They're made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Their monomer is called a nucleotide. And their purpose is to store our genetic information, the code of life. Two examples are DNA and RNA. And we store these codes in 4 letters, A, T, C, and G of our nitrogen bases. All right, that is all I have for you guys for this one. I will see you in class.